Hello, and welcome back to Corks and Quarter Notes. Today it is our pleasure to have our first ever episode with two guests. They each have a PhD in medical genetics, and we've asked them to have a chat to help educate ourselves and others about their field. They also have a brand new podcast called The Science Brief, where they interview experts in other fields about many topics of interest. So without further ado, I give you Dr. Maryam Hejazi and Dr. Vanessa Carias. Wow. What an honor it is to not have one, but two PhDs on a podcast. That is, thank you so much for taking some time to talk to us. First of all,
we're really excited to be here.
I think we'd like to start by hearing something about yourselves and your podcast. So for each of you, I guess let's start with Vanessa. What caused you to pursue your PhD in medical genetics and science in general?
Yeah, so I always loved science since grade 1, I guess. My parents always said I was into doing experiments and stuff, like the Mentos and the Coke bottle thing. So always loved science, knew I wanted to do some sort of science in undergrad. And then in second year, you have a genetics course. And I don't know, it was just like the first class, they started talking about evolutionary genetics. And then from then on, they touched on everything. And I was just so excited. It was also very interesting. And after that, I just picked every genetics course so I went to University of Western Ontario. And every genetics course they offered, I tried to take it. And then I was like, I love school. I guess let's do a PhD. And I did some undergrad research too. So that was kind of what pushed me towards that. I studied some bacteria. We were trying to understand one of the genes that makes a certain protein. So I did that first. And then I was like, I like research. I love school. Let's do a PhD. So then I moved out to Alberta where, first I was trying to see what kind of research I want to do. And I realized how exciting and how important it is to study genetic disorders. So I found a lab that studies a genetic disorder called Prader-Willi syndrome, met with the supervisor. She was really awesome, and then decided to do my PhD.
Very cool. And Maryam, how about you? How's your story similar and different to that?
Well, unlike Vanessa, as a kid, I wasn't into science. No one even thought I'll become a scientist. I was more into sports. But when I was in high school, we had biology course that had genetics. And we had this teacher. She was really good at teaching us genetics. And she used to give us really hard homework to find the answers. I guess that was the only course. I wouldn't leave it for the last minute. And I was very excited to find the answers. And I got good marks on it. And eventually I got into Bachelor of Molecular and Molecular and Cellular Biology. And I did my master back home. But it was my dream and passion to immigrate to Canada. So I had to leave all the school, come here, and I had to get a job because you need money. And for a few years I worked, but eventually I was in Ontario. Eventually I applied and I accepted at the U of A in a PhD program in medical genetics. And it's still some people surprised that I become a scientist.
Wow. That is... I can't believe someone who doesn't like scientists went into medical genetics, because to me that's just, oh my gosh, one of the hardest fields to... I know at the highest level, all the scientific fields are pretty even, but I feel like the floor of getting into biology in general is just...
There's a lot of memorization.
Yeah.
It is. Yeah. Especially in undergrad, so much memorization, so much.
What brought the two of you together? So you both started pursuing this. You went to the University of Alberta.
Well, we would hang out a lot because we were in the same department. So our labs were down the hall from each other. And we volunteered a lot together on the Medical Genetic Students Association, as well as Let's Talk Science. So that's a science outreach organization. You plan really cool, fun science events for kids. So we volunteered a lot. We were best friends. And then we graduated in 2019. And we had always talked about doing something like this, like some sort of science outreach, maybe a podcast. We weren't sure, but eventually we sat down and we're like, okay, we're done school. We have more time on our hands now. So let's do something. So the idea of the podcast came out. Took us like 6 months. I don't even know, maybe less, but it felt like a long time to think of a name and then a logo.
Oh, yeah.
So
And a logo.
yeah, the logo took us forever. But yeah, we just had a really good friendship and working relationship. So we're like, let's do it.
Yeah. Lunchroom helped.
Yeah.
Yeah.
Yeah, you had to stay sometimes long times in grad school for long hours, and we go to the lunchroom like later in the evening and hang out.
It's really nice.
And we also love learning about science. Like through our science outreach, I got to learn, some of the basic concepts, but it was like other cool stuff not involving genetics. So when it came to our podcast, we're like, oh, it gives us an opportunity to keep learning. And because we both love school so much.
Yeah.
So we tend to pick topics and experts that we're interested in, but not necessarily that we know so much about. So, one of our podcasts was aging, and that was cool because we got to talk to an expert in aging.
Yeah.
Mm-hmm.
So what would you say is the goal of your podcast, which is the Science Brief? You said that's the name you spent a long time trying to get to. Is there any special meaning behind the name?
Well, yeah, we talked about names a lot, and then I had this idea of like, I love the detective shows and books, so I was like, okay, what if like they have briefing room? So this detective with this whiteboard and go like, okay, let's solve this problem, answer this question, and they get some pictures, and I have this whiteboard, and I put like a question on it, and then do some research on it. So it was also Vanessa had some ideas of like, okay, then brief also is related to we don't go to detail. We try to make it understandable and fun for people and make science fun, and also at the same time, as Vanessa mentioned, because we love to learn, so we have to study and we have to do research on the topics, and we try to get out of our comfort zone and learn about new topics, meet new people like yourself. So that's how we got to the name, and then started like picking topics and doing few episodes.
That's really clever. What are some of your? Because I actually I read a lot in general, but I really love mystery as well. What sort of mystery authors do you like?
So when I was younger in high school, I actually read lots of Agatha Christie, like all the
so good.
Poirot and then the Marple, all of them. Like I was like stay up late and read all of them. And then later, like I watched few shows like Dexter, Lost, all of those mystery shows. I love them. Yeah. And True Detective. Yeah. So my mom used to tell me you watch too many serial killer shows. So, but yeah, I
I'm the opposite. I don't like any of those shows, but I do like a good mystery. So.
Yes.
Yeah.
I think, and I read, and then there were none. Pretty young, and I remember it just blew my mind.
Yeah, I turned out to be okay. So.
Yeah. Well, as far as we know.
As far as
the night is young.
Don't tell secrets.
So I guess we can start getting into the science a little bit. Can you explain exactly what is genetics?
Okay, so if I want to tell you like a definition, it's just like genetics is a branch of biology that study genes and heredity in organism. But to understand it well, I kind of want to take you to the time travel, go long time before even civilization. So I think humankind was always interested in knowing why my daughter have my eyes, my partner voice. So even like in small tribes, they must have been interested in the question why and how the kids or offspring get similarities to their family. But first, recorded speculation comes back to the ancient Greek. So there have been many theories about heredity and why offspring look like their families. For example, Hippocrates is a philosopher and called father of medicine. So he believed that all organs of the body of the parents gave off a seed during the sexual intercourse. And that's how like you get green eye from your dad and blonde hair from your mom. And then later, Aristotle actually believed in importance of blood. You might have heard like, oh, he's in my bloodline. So I think it came from that. So he believed that blood supplied generative material for building all parts of the offspring. He actually believed that semen and eggs were purified blood. And that's how this, he didn't know genes, but it's how this hereditary goes to the offspring, which I think it's very interesting because at that time they didn't have anything to do experimentation.
Not a bad guess.
It's a mechanism.
No, but it's just so smart to think about that, although most of them turn out to be not true, but still they have been used for a long time, and other scientists before or past philosopher theories to add to it. And then fast forward to 19th centuries. So imagine you walk into this garden full of peas, yellow, green, tall, short. So peas all over the garden. And this gardens belong to the guy called Gregor Mendel. It's not most probably because he loves peas in his food, but he actually started experimenting on heredity. So he started observing, crossbreeding few generations, like yellow peas, green peas, few generation, do mathematics, do stats. And he came up with theories and he published his work around 1866. And I quote, he demonstrating the action of invisible factors, which now we know genes, in predictably determining the trait of an organism. So he figured there are an invisible factor that determine the characteristic of offspring organism, but he didn't know what is this invisible factor. It wasn't until mid-20th centuries, Rosalind Franklin and Raymond Gosling produced the X-ray of diffraction pattern indicating a helical form for later called DNA. And then using the X-ray information, James Watson and Francis Crick demonstrated the molecular structure of DNA in 1953. So this was the first time that scientists know that, okay, there is a double helix and the building block of the DNA are chemical group called nucleotide. There are known to be 4 types of nucleotide. And these 4 types are genetic codes. It's like computer code. So these 4 types can become in different orders, make the codes of organism. And then the research goes on until 2001 that scientists could sequence the 3 billion nucleotide in a human. They published it.
So I have a few questions. One, how hard or easy is it to sequence, like you said, this DNA?
It's like as the time developed, it became easier, but still, I think it's more time consuming. I think Vanessa, do you know how? It's not hard right now.
So if I found like a lizard in the Amazon that nobody had seen, how long do you think it would take us to sequence that DNA? Is it?
Well, first of all,
days?
you if you
That's
have a
it?
lizard,
Well, if you think about COVID, they sequenced it in a matter of, I think it was
Months?
weeks,
weeks,
if
okay.
not less. Yeah. So,
I think,
and
yeah, I
that's
think
kind of like your
longer-
example. That's an example of a novel thing, right? That they didn't know the genome sequence of.
So another point I wanted to make, and this a little bit ties to when we were on your guys' podcast talking about quantum mechanics. You said that they were able to determine the structure of the DNA with a diffraction pattern by sending light through it. Is that right?
That I don't know enough. I think it was X-ray.
Right, okay. So you send the X-ray through it and then it diffracts. I just wanted to point out, you said that was about 1950. That is immediately after most of our quantum mechanical theories were starting to like settle down. So like the physics of knowing quantum mechanics is directly led us into being able to figure out the structure of a DNA.
Yeah, because it's very interesting. There's a movie, I forgot its name. It's about actually the Rosalind Franklin and Watson and Crick, and how the X-ray that they did, she did, or with Raymond Gosling, had essential role in for Watson and Crick for finding the molecular structure of DNA. Like they have been doing that's if I find the name, I text it to you. It's very interesting movie.
I'd love to see
And
that.
and if it's the same that like it was at the same time at the quantum mechanics, definitely physics helped
Right.
genetics.
Very interesting. And I thank you for mentioning Rosalind Franklin because she is often not properly credited for that accomplishment.
Yeah, no,
Yep.
it's very interesting movie. Which is interesting that it was. I didn't know that it was at the same time of the.
Yeah, just right after all those principles were kind of laid down in what we use today. So that I thought that was pretty neat.
That's neat.
So broadly speaking, whenever you have a sequence of DNA, understanding it lets you how does it let you interact with the organism?
So if you understand the sequence, you can, so it's kind of like reading a book, right? So the different letters, the base pairs, make words, which is protein, and then those proteins have different functions. So it's kind of easy actually, because a lot of genes have a start codon, so like a code that tells it this is where you need to start reading for the protein, and an end codon. So you can understand the different words in the genome, which is like the different proteins, and then each protein has a function. And through like using a lot of software that is beyond my knowledge, but you can like put the sequence in and see the different proteins that can come out compared to other organisms that may have similar sequences. So it's a lot of like comparing to what we already know, and a lot of that has come from research. Like if I think about my PhD project, we were trying to understand the function of this gene called MAGEL2, and it takes research like that to then you can be able to understand maybe families of genes, and then each piece like that you put it together. It's like putting together a puzzle. So you really need the sequence to even know what the gene does, or what kind of protein it makes, and then how those work together.
So whenever you have a piece of DNA that codes for just a single protein,
or a gene,
and
or
then,
not a DNA, right?
Right. So it's genes code for proteins. And then you have like a group of proteins that work together to do something, and then
To make
those
a
work
phenotype,
together, and then you get us, more or less. Is that how that works?
Yeah, that's exactly it.
Okay.
There's some genes that don't code for proteins too. This is one of the misconceptions Maryam and I were talking about, what used to be called junk DNA. And they would say, oh, it doesn't code for a protein, so it doesn't do anything. But now, obviously, as research progresses, we're realizing how important this so-called junk DNA, which it should no longer be called that probably, but they do have functions. So just because something doesn't code for a protein, it still can have like regulatory function and like have an effect on the other part. They're there for a reason, essentially.
So you said regulatory function. It wouldn't code for something as complicated as like behavior, right?
No, that's more like regulating the gene itself. So like gene expression. Yeah.
Okay.
Can you give an example of a regulatory function? I'm not quite.
Well, that takes us into, we can get into epigenetics in a bit, but the example I can think of right away is methylation. So it's a chemical modification, and that is something that attaches to the DNA and makes it harder for the DNA to be transcribed.
That sounds like the opposite of what DNA wants to do, generally speaking, right? Or
Well, not
No.
always.
okay.
Not always. So
You want the genes on.
yeah, a good way to think about this is all our cells have the same genome, right?
Right.
So our skin cells, our muscle cells, we all have the same DNA, but they don't need all the genes expressed, right? So the skin cell and the brain cell need different proteins to function.
And so they inhibit the ones they aren't using?
Yep, exactly. The ones they don't need in that cell type, they're turned off. And a big way that they do that is through this chemical modification called methylation.
Very
Wow, that's
interesting.
cool.
Yeah, I had no idea about about that. That's very interesting.
So do we know how much of the sequence is coding for proteins versus these regulatory stuff?
I've heard 1% coding, so I think,
Yeah.
like Maryam said, it's
So
not
only
really.
a tiny
It's not
percent
really
of your DNA is for
not
making
coding.
proteins?
Like only 1% is coding or 2%. I'm
Yeah.
not sure. I think there's a debate, but the rest is non-coding.
Right. So if I got this right, if DNA altogether is just like a manuscript of what you're supposed to do, like in an entire book, the cells from the brain only use page 60 to 80, and then the cells in your stomach use the first 10 pages or whatever, maybe page 14, but they like burn out the rest of the book or block it out and get out.
They still keep the rest of the book.
What's
They
still
just don't read
there?
it.
Yeah,
They
they're still there.
just
It's
ignore
just,
it.
it's just
They put a
silent.
little tab right there.
Yeah, they're sleeping. Yeah.
All right, very cool. What about genomics?
So it's the full study of your whole genetics.
Oh, okay. So instead of like the bits and pieces kind of as a whole.
Yes.
Yeah. So an example, let's say I say I study genomics. It's kind of versus genetics. So with genomics, I'm studying the whole organism's genome to understand what it does.
Right.
Whereas genetics, when I said I was studying MAGEL2, I use the word genetics. So that's kind of the
It's
difference.
just a little tiny bit of the DNA, and you're seeing
Yeah.
how it affects
Yeah.
the large
After
scale.
sequencing, it became more popular, genomics. After like more organism got sequenced, it became more popular.
Well, with advances in bioinformatics, right now we can look at the whole genome at once, whereas before you have to isolate one gene at a time. Now
Oh my gosh.
now you can just look at the whole genome. So
That's
it really is, yeah, it's really an advancement of science that and computing science that helped.
Yeah, I bet it does actually.
I would not want to do that with paper and pen.
Bioinformatics is, or it's itself, it's a field, and I suck at it at least.
Yeah, that's beyond us.
Don't worry, I'm pretty bad at it too.
Okay, don't ask any bioinformatics.
So still on genetics and genomics, I want to touch on mutations. What is a mutation? Does it happen at the level of single genes or groups of genes?
So I said that their DNA has 4 nucleotide, and it's just like a code.
This
So
is like your A, T, C, and G. If I remember from biology.
And they can put in different configuration and become a new code. But like any changes, it can be deleted, it can be translocated, it can be changed. Of single nucleotide is mutation. So we can have more than one nucleotide, still is a mutation, but one single nucleotide, any changes, we call it mutation.
Okay.
Some of the mutation are silent. It happens most probably during cell division all the time. Some of them gets fixed, some of them doesn't, but nothing happened. We don't even know about it. It's process of like genetic replication during that happens. So it's not harmful and it's not useful. It could be in one gene, in one sequence, and
Makes no change.
Makes no changes in protein.
Okay.
But
So
some of them are harmful. Lots of genetics disease like Prader-Willi, lots of like ALS, like disease,
cystic
fibrosis, and then not all of the mutation are harmful too. So some of them silent, some of them harmful, and then actually there are useful mutation. One example is very interesting. There are this population, HIV resistant population. It's mostly Northern European. There has been lots of research on them. So it turned out they have a mutation in a gene that become an immune cell later, like a protein. And the mutation caused that the HIV virus cannot enter to the cells and then replicate and infect the cells. As a result, the individual does not get HIV, and it causes HIV resistant mutation. So this is kind of a useful mutation. So there has been like research on it. I'm not sure it's exactly like where it comes from, but it says it's mostly in Northern European population.
So this might seem like a silly question, but I'm imagining a strand of DNA, and Are there perhaps like pressures from outside influences that make parts of the DNA more or less prone to mutation?
Yeah, like aging is one of them. So we can talk about later. So as we get older, it gets error happens more, and we have protein that fix these errors. And then as they get older, they become more mistaken, and the odds of harmful mutation gets higher, and eventually might get to cancer.
Or carcinogens, right, Miriam? Like
Yes.
with if you're exposed to carcinogens, it can trigger your cells to replicate faster, right? So that's another example of environment changing how your cells work, and you can end up with cancer.
And so we have proteins that look for mutations and attempt to fix it.
Mm-hmm. Yes.
When the cells replicating. Yeah.
Replicate. It's their job to fix the mutation.
Like we have it's DNA repair, they call it, and it's a mechanism that helps to fix those mutations. And sometimes if they escape that, that's when you can have mutations that can cause disorders.
This is going back a little bit to when you were explaining the history. So I recently read on an Origin of Species, which was written, I think, a bit just around the same time as what you were talking about with Mendel and his pea plants. I remember reading the Origin and being blown away at how well it seemed he understood the process of how genetics works without knowing anything about DNA. Why did he and Mendel talk at all? Did they not cooperate? Why did it take so long for Mendel's
It is quite
stuff
interesting.
to-
When I was doing my research, I think I wrote it down somewhere on my research. They were at the same time.
Yeah.
They were at the same time points. And then I did research on it that for my curiosity, if they had they had no interaction. They didn't know about each other research. Most probably if they knew, it would be like, oh, yeah, I did whole research on it. I got disrupted so much one night. I was just like, why this guy didn't know? I guess it was just before the media and stuff.
Yeah.
So yeah, they were at the same time, but Mandel has did more experimenting on heredity, and then Darwin was more on natural selection, which is genes, but
Or evolutionary genetics.
Or evolutionary genetics, but they did not connect it, which is unfortunate.
Evolutionary genetics as opposed to what? Or
I guess we call it Mendelian genetics, where because it's named after him, but where he was following traits, whereas I feel Darwin was more looking at how they've changed over time. So yeah.
Gotcha. Okay, so I guess we can maybe go ahead and dive a bit into epigenetics. Maybe if you could just explain what epigenetics are before I go any farther.
Yeah, definitely. So epigenetics, we kind of touched on it, where it's it controls gene expression, or how much it's expressed. So what I kind of want to use an analogy with the book again, where you have your DNA sequence and it's the words. And then let's say you are writing a book. So those are the words. That's your DNA sequence. Then you send your book to an editor, and the editor, they mark it up. They put a bunch of cross out this word. So then epigenetics is all these markups or these chemical tags that then can turn on a gene, turn off a gene, or can control how much of the gene is expressed. So then how much of the protein is made. And that's pretty much what it does. Epigenetics are these tags. And one of the example is the methylation, but there's a lot of other modifications that can happen on those histones. So that's, again, the protein that the DNAs are wrapped around. So methylation turns off transcription. Another one is acetylation that turns on transcription. So that means that these modifications open up the DNA a bit more. And then those machineries to turn on can get in there and actually turn on the gene. So epigenetics are not a change to the DNA sequence at all. It's a change to the modification of that. So no change in sequence, but change on the levels, and it kind of turns it on.
What does it mean for a gene to be expressed?
So it just means that it's turned on now, and the machinery, so that word transcription, so going from DNA to RNA, there's a bunch of proteins that do that. It's a whole process, but for something to be turned on means that that machinery got to the start codon. So that's, remember, the beginning of the gene. It recognizes that, and then it starts to read it. And when it's reading it, it makes the RNA. So that's transcription. Then from there, there's different set of machinery that takes the RNA to protein, and then that's called translation. But epigenetics is working at the opening up the DNA for it to be transcribed into RNA.
And we talk about transcribe. Would a good analogy be like taking this book of DNA and copying it over onto another book for like a new cell, or is that?
No, that's more like replication
Okay.
when it's I think a good analogy for transcription would be taking a bunch of letters, maybe they're spaced out a little bit, and putting them together so they make sense. So like, let's say THE, they were spaced out. Now you're bringing them together, it makes a word, which is the RNA. And then from there, you make sentences, and that's the protein. And then things are starting to make sense, right? And now you have a function. The sentence is the function.
So so no actual change happens to the DNA itself.
No.
And so is that just caused by environmental factors, or?
No, so epigenetics is just, it's a modification on the DNA. But let's start with, can you inherit it?
Yeah,
Let's go.
I guess that's really what I was asking.
So if you have your sequence and you have these modifications, you can pass some of these down to your offspring. But what happens when you have the egg and the sperm get together? What happens is something called the reprogramming, and that can get rid of it. It's like a fresh slate, and it gets rid of some of those modifications, but some of them do stay. So yes, they're heritable. Some of them are, some of these modifications aren't. And I think there's still a lot of research into understanding exactly what and why things are being inherited and which ones aren't. But a really easy example, or a really cool example for me, it's because I studied Prader-Willi syndrome. You have a part on chromosome 15, so it contributes to two different disorders. So this is called, first, it's called genomic imprinting. So this is where you have something from your mom or from your dad turned off to begin with, and that's normal. We all have it. But if you have the one that's turned off, so the chromosome 15, this section right here, if it's turned off from your mom, because that's how it is, but then you get a deletion or some mutation from your dad in that region, then you get this disorder, Prader-Willi syndrome. So your mom's is already silenced through that chemical modification called methylation. So that's already silenced. Now, something happened when your cells were replicating, when the egg and the sperm came together, something from your dad got deleted. Now you have no copies of that gene. So you get Prader-Willi syndrome. So that's an example of something that gets inherited. But if it messes up, then you end up with a disorder. So that's a really interesting example, because on the flip side, genes right down from that section I was just talking about, it's normally silenced in the father. So that's okay. But if you get a deletion in the mother, then you get a disorder called Angelman syndrome. So it's this whole special region on chromosome 15 that if the genomic imprinting, it's fine, but then if the other chromosome is deleted, you get two different, they're actually pretty different genetic disorders.
Yeah. Wow. This might be a dumb question, but how can epigenetic information be herited when it is not in DNA? Because my understanding was that DNA is the information that gets passed to us. What is it? Its own thing? Like, does it have a code of its own? I'm having trouble.
I would say it's attached and coming along for the ride. So you have your DNA, you have the modification. You're getting, when you have an egg, that all the meiosis and all the processes that do genetic recombination. What happens is that piece of the DNA has a tag, and it's just coming along for the ride. And then the part that I think we're still trying to understand is why some of them get erased and some of them stick.
So it seems like it's picking out the bits and pieces that it wants to come along or not reset in the reset process?
Yeah, maybe things that are important to evolution. This actually brings us to the environment thing, which is really interesting because environment really can either turn on some modification or turn off. So things like your diet, if you're obese, physical activity, tobacco smoking, alcohol consumption, uh, stress, all of these things can affect your genes essentially, and they can affect these epigenetic modifications and either like help them along or deter them from turning on. But a good example in mice, it's kind of interesting how there was one study where they exposed the mouse to something, or sorry, it was a rat. So they exposed the rat to something that they like to smell, but then they put like a shock every time he smelt this, and they conditioned him to not like the smell anymore. And what happened is there were some changes in the neurons, epigenetic changes in the neurons, that then got passed along to all the offspring. And you can see that modification, or you can see that result. So they would expose those kids to the smell without the shock, the offspring, and they didn't like the smell. They were already kind of in tune with what the dad had gone through, and that was because some epigenetic changes were transferred, or I guess inherited by the offspring.
That is
So
so
they
strange.
they learn something from their parents' experience. That is insane.
That sort of reminds me of the Lamarckian idea of evolution. You know, that like the giraffes stretch their necks so that the offspring, now they have longer. It's almost that sort of same kind of thing, but a little more scientific.
But within the cell. So
Real.
yeah, it's really interesting how our environment, like there's so a lot of twin studies too, right? Where twins are either raised the same or raised in completely different environments, and they either there's a lot of different studies, but a lot of it shows that even their personalities can tend to be the same even when they're growing up in different locations. So they say that that can be due to epigenetic factors, but also DNA. So getting back to genetic sequence, that can actually mean a lot when it determines things like personality traits. But then there's whole nature and nurture, which also plays a big role.
Yeah, I think one of the topics that probably is at the forefront of many people's minds in general when you're thinking about genetics and such is aging. You had a podcast on the Science Brief about aging. I thought it'd be interesting to talk about the genetics. You've already touched on how the cells get older and they get worse. I guess first, would you consider aging a sort of genetic sort of disorder, or purely environmental, or 50-50? Where do you guys place it?
So yeah, like that aging episode, it was just very interesting and I learned a lot. So Dr. Nir Barzilai is one of the I don't know, top scientists in aging research. So we learned, yes, aging is a disease and there's theories, and it's a genetics involved in aging. So as I said, there are biological process of aging, like cells gets divided. It's like a machine. So when machines after a while, it's just like accumulation of various type of damage result in cancer and other type of diseases. But most people does not consider that as a disease because, well, it's like, well, we got older, but all the disease arise from aging is because that damage accumulation.
Would it be accurate to say so? This, because it's almost a philosophical question asking if aging is a disease or not, because
So philosophically, I wouldn't call aging a disease. I think it's just what it is. Our cells divide, we get older, things happen, and I personally don't think I call it a disease, but it doesn't mean that it doesn't warrant its research, and why not try to see how we can extend life and make it healthier and make it happier for people. I just I wouldn't call it a disease, but if you Google, it does fit all the disease, everything that you need to call something a disorder or disease. It fits the requirements. So I get why it's called a disease.
Isn't one of the requirements to be a disease is that it needs to be abnormal or something to that nature? I mean, we don't have a word for the disease of people who get headaches or You know what I mean?
Migraine, I guess.
Or people who need to sleep, or I guess.
Narcolepsy.
Well, no.
I think
Like
it's
you and I need to
biological.
sleep regularly. We can't
Oh,
get away
yeah.
without sleeping. We're kind of condemned to sleep.
Oh, you're kind of saying it's a natural process of the human
Right.
body.
It seems like everyone ages and everyone needs 8 hours of sleep to or something like that. You know what I mean?
Yeah. Maryam, what do you think?
I was just gonna say in a biological term, called disease because eventually you die from a disease or multiple disease. Then if there's no genetics involved and you get a cancer, it's the cancer is related to your aging.
Mm-hmm.
Yeah, I guess it's a hard philosophical question, but
well, is the
It
need
is.
to drink water or you die, like, would you think of that as a disease?
Well, if you guys want to know, I barely drink water. Like I think some days I have no sips,
Oh, no.
and I'm fine. So
Maybe you don't
I'm
need to. I don't know.
I talked to a doctor once, and they said I probably get enough through fruits and coffee.
That's very true.
That's how I survive.
Right. But so Vanessa, you say you think it is so you wouldn't take the never age pill that just completely cures aging, or
Is everyone else taking it? Can the world? Can
To
the world
live
system-
longer would be nice. To live forever.
yeah,
Yeah,
if
about
I
that
tell me
one.
I'll be healthy, relatively healthy.
yeah.
Yes, but it's not a scam because there's lots of scam on aging disease, and it's been clinically like approved and like it's been researched. I take it.
Yeah, maybe.
Yep.
So at least for me, whenever I'm thinking about just like life in general and what it means to be and those kinds of questions, like one of the facts that's given to me is that I'm only here for just a little bit, right? And so like on this philosophical note, it seems important to like be aware of that because it makes you're just like, well, I'm only here for a little bit, which means I can only do a few things. What are those few things I can do, right?
Like make the most out of
Yeah.
the time.
Right.
Yeah.
Or if you had forever, you're just like, well, I could get to that,
Yeah.
whenever because I have forever, right?
But if you think about that, like 200, 300 years ago, like the average of age most probably was like what, 40, 50 years old, like
Right.
Younger, probably. Yeah.
before.
Something's doubled.
But right now is what, like I am not sure, but I'm sure it's more than 60 or 70. So that also
Science is
that
doing something. Science and medicine, and they're doing something to extend our life. And who's to say in 50 years it doesn't extend another 20, right?
Right.
Yeah, well, and then in that respect, we certainly are treating it like a disease. We're actively trying to find ways to fix these problems.
Yeah, that's true.
So, and we're not actively looking for ways to reduce the amount of sleep we need, I don't think, as far as I know.
no. Also it's because of
I don't think so.
economy and like because
Maybe energy drinks. I feel like that's a way to reduce. Actively, whoever's like doing research in energy drinks is actively
Maybe.
trying to reduce our sleep.
or like
So-
both
So to end, I wanted to ask you guys more of a question for myself, or I guess anybody who's going through the experience of academia. I wanted to know what y'all thought about grad school, what your expectations of what grad school is going to be. Looking back, what are some of the things that were unexpected or that you wish you have done?
So for me, I don't think I knew anything that I was getting myself into. I was the first one in my family to go to definitely grad school and one of the first to do a bachelor. So I didn't really have anyone in my family that I could ask, and none of my friends because we were all in the same boat, really. So honestly, I had no idea what to expect. And when I got in there, it was hard. Like the 5 years, I think 5.5 years, were hard. And, I learned a lot about myself, and I wish that I would have been more organized. I wish I understood the process and could have done better. Like now that I've been through it, I would have taken better notes like one of my biggest regrets is not documenting everything exactly in my lab notebook. Like I just wasn't really good at that, and I think that would have made my life easier when it came to the last 2 years where, like, you know, you had to make sure everything was good and follow up on experiments from the years before. So one of my biggest things is like get organized and do it fast because it makes your life easier. But yeah, no, it was a hard experience. Anyone who goes through it, I'm very impressed because it's, you know, it was difficult, and I enjoyed it. I loved learning about research. I loved when I had to present my research. Like I was always worried with presentations, but by the third year, I was like, it was cool to talk about your research, and I also met the families and the children of the disorder in my second year, and I think that's when I was like, wow, they're like excited for me to talk about the littlest thing, right? Like I told you guys, I was studying one gene. What does this gene do? But to them, that's their whole world. Like if we could answer that, even if it takes 5 years, 10 years, maybe 15, 20 years, they're living with this disease for their whole lives. So if they can even get a little bit of an answer, then my 5.5 years was worth it.
Right. So you mentioned it was harder. Was it like a heavier workload, or was the material more technical?
I think it was the workload and the pressure I put on myself. Like I was in the lab, I don't know, I was there from like 8 till 9 p.m. most days, and then I was in on weekends, and it was like felt like sometimes I was getting nowhere. So yeah, it's just, experiments take a long time. I think you need to know to have patience with yourself, and also maybe seek out help more when you need it. Like that was one of the things maybe I wish I would have asked my supervisor more for help, or, you know, there's a lot I wish I would have done differently, but at the end of the day, it's just kind of don't put too much pressure on yourself. One of the advice that I'd give anyone is do other things outside of just, like, if it's a thesis or program or course based, doesn't matter. Do things outside of that. Like what brought me so much joy was doing the Let's Talk Science, and I was the president of the Medical Students Association. Like those things were so exciting to me, and also helped me build my resume to get a job after. It wasn't just, I didn't just go to the lab and that's all I did. So I think a big part of it was like making friends like Maryam, and like making sure you got other skills. Like your PhD gave, like, think of a skill, you can figure out a way to explain how your PhD gave you that skill, but also it's good to have like other experiences too.
If someone asks me like how to build endurance, I say go to PhD. Like you get resistant to almost everything in life. So yeah, and I was also in a new country with like new culture and new language. So it was just, but it was, people around me was very helpful, like the department we had. So I think people is very important. So find, as Vanessa said, find a bunch of, I don't know, like, or one even friend that you enjoy doing stuff outside. Then like those lunchroom breaks, or I don't know, like I would say supper breaks, helps to get through the failure of the experiments. And then all those stuff, Vanessa, I wish I was more organized. I wish, but I know if I go back, it's the same. So there's no free will, I feel.
But you know what? Like now I have this notebook for work, and I document every little thing. And guess what? My life is so much easier now. So that's like number one rule.
That is one of the things coming back to school that I forced myself to do, was be organized. And so
it's huge.
It really is.
Thank you so much. And like you said, those few years you spent learning about that one gene, that might be the world of difference to people suffering now, and who knows how many people in the future might be one day helped by that one contribution. That's amazing. Thank you so much. And thank you guys for coming to talk to us. Do you have any final things you'd like to say?
No, thank you for having us. We really enjoyed this like very informal talks. I really liked it, and yeah, thanks.
I think I had my mind blown 6 or 7 times so thank you for that. And everyone listening, be sure to go check out their podcast, The Science Brief. Is it Science Brief at Science Brief on Instagram as well? And
at The Science Brief.
At The Science Brief, okay. And we'll be sure to have the links to that and to all the stuff below, and we'll even link some of the things we discussed, some of the papers. All right. Well, thank you guys very much.
Thank you.